We are very lucky to have two wonderful presenters today joining us from Riot. And so our first presenter is Patty Suchimit. And Patty received her doctorate in stem cell biology and immunology from the University of Oxford in 2016, where she investigated the differentiation of immune cells from stem cells for immunotherapy. She then moved to Toronto to pursue a postdoctoral research fellow at the Hospital for Sick Children, where she was engaged in the discovery of novel drugs for the treatment of glioblastoma, an aggressive brain cancer. And so Patty currently works as a scientist in the Translation Immunology Group at Blue Rock Therapeutics, a Toronto-based cell therapy company with a mission to develop regenerative medicines for intractable disease. Next slide. And we also have Kinjal Desai joining us as well. And Kinjal is also part of Toronto's Research Information Outreach team at Sick Kids Hospital. And so Kinjal is a senior postdoctoral research fellow at the Hospital for Sick Children, working on developing a novel treatment for medulloblastoma, the most commonly occurring malignant brain tumor in children. She is also an instructor at the University of Toronto for the School of Continuing Studies and has guest lectured at the University of Toronto Molecular Genetics Department. And so I think with no further ado, I'm very pleased to introduce both our speakers to deliver our presentation for today.
Thanks very much, Anita. It's a pleasure to be here. And I'll just kind of get us started, do a little bit of an intro about our organization and just a little bit of answers, and then Patty will take us through the immunological aspects of this talk. So we are the Toronto Research Information Outreach team. We're affiliated with the Canadian Cancer Society. Over and above doing these kind of sessions, Patty, I think you might have to click to the next point. Over and above doing sessions like these, we also do high school talks, and then we post blogs on our website, torontoriot.com. And on the left, see a couple of our more recent. So we do try to stay very topical and discuss issues that are really popular and questioned in the media. A bit more about who we are. We are cancer researchers who volunteer with the Canadian Cancer Society. As I mentioned, our basic goal of this organization, Riot, is to share our knowledge of cancer and highlight current research. We're not licensed medical professionals, and so just a bit of a disclaimer, and we're also not qualified to give medical advice, so that's something that's best left to your professional oncologist or a doctor. We're really happy, though, to discuss any questions about cancer biology, research, whatever you see up here on these slides, or anything besides that. So today we're going to talk about immunotherapy. So we'll begin with a little bit about cancer and cells in general, and then move into what the immune system does, key players in the immune system, and then sort of have a discussion about cancer and the immune system, how they kind of interact and whether. Immunotherapy is kind of the big topic, and it is harnessing the immune system for cancer therapies. So spend some time discussing that, the different types, and talk about a few case studies, including a clinical trial, and then plenty of time for questions and discussion, I hope. And also feel free to interrupt, or sorry, I think as Vanita mentioned, yeah, just sort of put your chat in whenever you question, and then we'll definitely try to get to it at the end. So to begin, let's talk about how the study of cancer rests on our understanding of normal cells. So we have a normal cell on the right, sort of depicted as a cartoon, and human tissue and our organs are sort of composed of cells, which are the basic unit of life. And so all cells perform basic functions such as respiration, this is to get oxygen or utilize the oxygen, breathe, power generation, and so like any organism, the cell at a single level also needs to do all of these things to sort of work properly. So power generation is to sustain itself. Of course, it needs to remove waste so it doesn't up in the cell and sort of hamper its activities. And then cell division, of course, is to maintain itself. And so these are all the functions performed by every single cell in our body. Cells, other than the basic that I just described in the previous, it also has specialized functions. And so not all cells in our body are the same. Cells do become specialized according to their function. So on the right, we see a picture of a skin cell, and the skin cell has a number of different components. And they're also known as melanocytes. And so melanocytes are the only types of cells, or one of the few types of cells in our bodies that make melanin, which is the sort of pigment that protects us, our cells from the sun, or violet rays, and sun damage. And so not all cells in the body, though, would need melanin. And so melanocytes have become specialized to make melanin and sort of function in that way. Our different cells, of course, work together and communicate with each other. Cells also have a system of checks and balances to make sure everything is working smoothly. And this is very, very important, because when things aren't working smoothly, the checks and balances will kick in to try to fix that and kind of repair things. So the way that normal cells work is to follow very specific and predictable patterns. So sort of as we described in the previous slide, cells have basic functions that are shared by all cells. They also have specialized depending on what kind of cell it is, where it is in the body. And of course, it's also very important that cells respond to signals from the environment. And this is key because the environment keeps changing. And so it makes a lot of sense for cells to sort of interact with the environment along interacting with each other. Important things about normal cells is that they know when to stop dividing, when they're either too old to maintain their cellular integrity and they're sort of starting to experience a lot of mistakes in the cell division or any other processes, or if they just got damaged and they're too damaged to be properly repaired, they do know when to stop dividing. And in fact, when they get too damaged, then we can move on to the third point, which is that they will undergo something called a programmed cell death, apoptosis, and they'll maintain the healthy life of all the cells and tissues in it. So one of the major differences, as you can probably guess where I'm going with this, is that cancer cells sort of start to behave abnormally compared to the normal cell characteristics that we just described. So some cancer cells do not know when to stop dividing. And so on the right, we see a picture of different kinds of skin cancer. And so whereas we had seen pictures of the normal skin cells and peptides, this is when skin cancer starts to develop, and you can see that it starts to look quite abnormal. So they don't know when to stop dividing. They cannot sense the environment. They sort of grow, they don't undergo, and this is a very, very important point about cancer cells. They, of course, grow where they shouldn't. Sort of an example of this in the lab is, so sort of Patty and I both work with cells in dishes, and we have observed normal cells as cancer cells in the dish and lab. And what we notice is that when we grow normal cells, when they reach the edge of the dish, or they've used up all the food that we've kind of fed them to grow, they stop growing. But cancer cells don't do this. And so the checks and balances has failed in the cancer cell. And even when they run out of space in the container that we're growing them in, they start to grow on top of each other and sort of don't seem to care or go through the normal inhibitions that the normal cells experience. And therefore, cancer is rapid and uncontrolled cell growth. And the abnormal characteristics that are acquired by the cells can actually have been depicted in this way almost 10 years ago in sort of a landmark scientific publication. These are known as the hallmarks of cancer. And so we'll just kind of go through some of them. Of course, we're going to discuss the immune aspect of it in much greater detail, but a couple of different ways in which cancer cells kind of grow abnormally is to sustain cell division. So there's these checks and balances. There are different kinds of signals. The growth suppressor are one that stops cells from growing sometimes. They evade those. They sort of induce blood vessels to form in order to get some nutrients and keep growing. The DNA starts to get many gene mutations. They resist cell death. They even sort of channel the cell's energy processes and production in order to benefit themselves and to grow, sort of to the detriment of the neighboring normal cells. And so these couple of ways, and then of course there's more in which cells acquire these abnormal characteristics or these halls of cancer, and this has been seen across all cancers. The immune system do. We all know that our immune system sort of protects our body and our sort of cells from invaders. So the invaders could be virus or bacteria, different kinds of parasites. Tumor cells, even though they come from ourselves, they appear different from us. And so they are also considered to be an invader immune system. And so the immune system is sort of a defense in our body that's kind of keeping away through different mechanisms that I'm going to talk about very soon, sort of keeping away these invaders or these foreign bodies in us. And so that's kind of a brief explanation of the immune system. Of course, there's a lot of different aspects of the immune system, and it works in a very coordinated and complicated way with a lot of different players in it that sort of work together to keep everything functioning smoothly and to prevent us from getting sick more than we already do. So I think we have a small short video, just a couple of minutes or a little bit more, that kind of very nicely describes how our immune system works and has some really wonderful depictions of that. So we're just going to play that for you and then keep talking about the immune system in cancer.
A mosquito lands on your arm, injects its chemicals into your skin, and begins to feed. You wouldn't even know it was there if not for the red lump that appears, accompanied by a telltale itch. It's a nuisance, but that bump is an important signal that you're protected by your immune system, your body's major safeguard against infection, illness, and disease. This system is a vast network of cells, tissues, and organs that coordinate your body's defenses against any threats to your health. Without it, you'd be exposed to billions of bacteria, viruses, and toxins that could make something as minor as a paper cut or a seasonal cold fatal. The immune system relies on millions of defensive white blood cells, also known as leukocytes, that originate in our bone marrow. These cells migrate into the bloodstream and the lymphatic system, a network of vessels which helps clear bodily toxins and waste. Our bodies are teeming with leukocytes. There are between 4,000 and 11,000 in every microliter of blood. As they move around, leukocytes work like security personnel, constantly screening the blood, tissues, and organs for suspicious signs. This system mainly relies on cues called antigens. These molecular traces on the surface of pathogens and other foreign substances betray the presence of invaders. As soon as the leukocytes detect them, it takes only minutes for the body's protective immune response to kick in. Threats to our bodies are hugely variable, so the immune response has to be equally adaptable. That means relying on many different types of leukocytes to tackle threats in different ways. Despite this diversity, we classify leukocytes in two main cellular groups, which coordinate a two-pronged attack. First, phagocytes trigger the immune response by sending macrophages and dendritic cells into the blood. As these circulate, they destroy any foreign cells they encounter, simply by consuming them. That allows phagocytes to identify the antigen on the invaders they just ingested and transmit this information to the second major cell group orchestrating the defense, the lymphocytes. A group of lymphocyte cells called T cells go in search of infected body cells and swiftly kill them off. Meanwhile, B cells and helper T cells use the information gathered.
Hi, I'm sorry. I think it got paused by mistake. Let me just make sure that everything's fine. And we'll continue from where we were at.
cells and swiftly kill them off. Meanwhile, B cells and helper T cells use the information gathered from the unique antigens to start producing special proteins called antibodies. This is the pièce de résistance. Each antigen has a unique matching antibody that can latch onto it like a lock and key and destroy the invading cells. B cells can produce millions of these, which then cycle through the body and attack the invaders until the worst of the threat is neutralized. While all of this is going on, familiar symptoms like high temperatures and swelling are actually processes designed to aid the immune response. A warmer body makes it harder for bacteria and viruses to reproduce and spread because they're temperature sensitive. And when body cells are damaged, they release chemicals that make fluid leak into the surrounding tissues, causing swelling. That also attracts phagocytes, which consume the invaders and the damaged cells. Usually, an immune response will eradicate a threat within a few days. It won't always stop you from getting ill, but that's not its purpose. Its actual job is to stop a threat from escalating to dangerous levels inside your body. And through constant surveillance over time, the immune system provides another benefit. It helps us develop long-term immunity. When B and T cells identify antigens, they can use that information.
Okay, I'm going to stop it there because it's going to go on for a little bit, but I think that sufficiently explains the basics of the immune system. I love this video because I think it does such a good job of explaining, what the immune system does and gets across the complexity of all of the cells of the immune system working together to help protect our body against infectious pathogens. So I'm just going to recap here what the immune system does. If you remember, they in this video classified leukocytes, the major cells, the immune system that are responsible for protecting us against these nasty bugs and infections into two classes, phagocytes and lymphocytes. And this slide is just for recap purposes. The video has already outlined this, but it'll become pertinent a little later when we start talking about various types of immunotherapy. So phagocytes are the cells that are the ones that will go on the lookout. They're the sentinels of the immune system. They go on the lookout for anything that's foreign, anything that doesn't look right. And that includes actually cancer cells. Like cancer cells, some cancer cells will exhibit like kind of telltale signs, if you may, on their surface to say that, oh, something's off, like I'm not a normal body cell. So these cells are really good. They'll be able to go, oh, this looks like a bug, oh, that's a bacteria, that's a virus, that's not right, that's not supposed to be in here. And then they'll go and tell the T cells, the lymph and the other lymphocyte, they'll be like, okay, there's something wrong, you've got to deal with this. And then the T cells will then kind of produce chemicals to go and kill the bugs, search and destroy the bugs. And the B cells, once they the phagocytes signal to the B cells, and all of these cells work together. The T cells will also help activate the B cells. They'll produce lots of these antibodies, these little molecules that will help and neutralize the invaders, but also help the pathogen get recognized by other cells in the immune system. So it's kind of like a nice feedback, kind of all of the pieces fit into another, they feed back into another, they all help each other work. So it's kind of a complex system that it's amazing sometimes what evolution has managed to do. If you remember, Kinjal a few moments ago, described to you some of the various hallmarks of cancer and explained to you how cancer, a normal cell becomes cancerous and what is different between a normal cell and a cancerous cell. And one of the hallmarks of a cancerous cell is its ability to avoid immune destruction. So its ability to evade the immune system. Now, research over many, many years has shown that the immune system is actually actively able to recognize cancer cells. This is like a depiction of what goes on in the body, a very nice diagram based on real data. So this shows a T cell, this small cell over here. This is one of your lymphocytes, in the last slide, approaching a cancer cell. recognizing the cancer cell, making contact, and this contact, when it makes this contact, it basically, the T cell is secreting certain chemicals into the cancer cell that will cause the cancer cell to die. So it's attacking the cancer cell, and then the cancer cell is destroyed. So if indeed our immune system is actively, surveying the body and has the ability to recognize not just bugs like bacteria, viruses, or anything like inflammation from a cut, but also able to recognize cancer. Why do we still get cancer? Right? Isn't this the immune system job? It is indeed the immune system job, but cancer cells are very smart. Cancer cells have the ability to hide from the immune system. They've evolved mechanisms to escape the immune surveillance and escape immune response. And data has shown that if you have, if we didn't have the immune system, like we'd get cancers all the time. Like it would be like, mice that don't have the immune system are way more prone to getting cancer. So the immune system will get some of them, will get some of these cells earlier on that have changed and transformed into cancer cells and look different. But some of these cells manage to evade. And these are some of the mechanisms by which tumor cancer cells evade the immune system. So first of all, when a cancer is formed, if it's a solid tumor, there is actual barriers to the actual mass of the cancer cells. So that is like, just like a fortress that the cancer is building, that's making barriers to the immune system to come in. So it becomes harder for the immune system to come inside that space. In addition to that, for those immune system cells that do come into that space, or in blood cancers where it doesn't form like a physical barrier, the cancer cells do a very good job of depriving the environment of the nutrients that the normal cells require to function, the immune cells require to function. So within that environment, the cancer cell is depleting nutrients, and the immune cells either cannot live or won't even come into that space, or even when they do come in, they change. They are metabolically different, and then they change from being an active to an inactive cell. They also are very good at hiding in general from the immune cells, because the immune cells are trained, right? They know what is self. They've trained to recognize your body cells, and mind you, the immune system doesn't always get it right. There's autoimmune diseases for a reason, and there's infections that go awry that the immune system doesn't recognize, but more often than not, the immune system does get it right and is very good at recognizing cells that do not belong in the body, or any pathogens that don't belong in the body. However, in the case of cancer cells, they are self cells. They are own cells. So in that way, it's harder for the immune system because it looks like one of us. But then a lot of cancer cells, because they've changed, they have mutations that cause them to change, they exhibit telltale signs. Some of these cancer cells have evolved and learned to hide these telltale signs. So they hide it, and the immune system cell comes along, and the immune system cell is looking at them. It's like, okay, this is just one of us. So it goes away and it doesn't do anything. So basically that's like the loss of recognition signals, and it's able to hide from active immune system surveillance. And another really good way that the immune system, a really smart way, I won't say good, that's the wrong word for it for sure, a very smart way that the cancer is used to evade our immune system is by giving the immune system suppressive signals. So even when the immune cells do actually recognize that this cell is our own cell, but something's really off with it. It's not well. It's not like it's a cancer cell. Let's get rid of it. It's still not able to function. The cancer cell is actually suppressing it. It gives these signals to the immune system, says, nope, you cannot kill me. Stop working. And then usually that leads to the immune cell, get changing from an active to an inactive form. So as you can see, there is this interplay between the immune system and cancer cells where there's constant equilibrium. The immune system is trying to detect the cancer cell is finding multiple ways to escape. And immunotherapy has come to be a field so that we can reactivate. How can we reactivate these immune cells to recognize the cancer? How can we help the immune cell get over all of these innovative ways in which the cancer cell is hiding from it or is stopping it from working? So as many of you have heard, like immune system has become the immunotherapy has become like a really hot area of research in the last few years. And these are some of the front pages, the covers of the biggest journals in the scientific field. And immunotherapy has been kind of front and center for a very long time, and for good reason. Like it's the first time in the history of cancer that we've ever seen complete remission in some patients, and that is as a result of immunotherapy, which has brought a lot of excitement and research into this area. And there's currently so much research. Like the amount of abstracts have like quadrupled in the past 10 years, and so many clinical trials. I'll show you, like there's like 2,000 clinical trials going on that involve immunotherapy. So that depicts like how much of an interest and how much promise this field has, but there's still a long way to go. We've come a long way, but I think we're just reaching that point where we are starting to get a better understanding of the field and a better understanding of the immune system. So what I'm going to tell you is actually, even though immunotherapy seems like a new field because it's just, caught the public's eye and caught a lot of scientists' attention as well. Immunotherapy has been around for a very long time. In fact, it's been around since the 1800s, and a lot of the research that we do today rests on some of the early observations by I was going to say scientist. He really was a physician in the 1800s. It's called Dr. William Colley. He had a patient that was very young, like I think she was in her 30s, and she died of sarcoma. And he just couldn't understand how aggressively, this disease took over her. This young patient who was really healthy otherwise passed away really quickly, and he was very upset. And he was like, what can he do about it? So he devoted his time, reading all of these texts, learning more about the disease. And he's like, how do I treat this disease? And what he observed was that a lot of patients who caught bacterial infection, streptococcus infection, actually survived some of these sarcomas. And it was through this observation that he developed a hypothesis that, maybe if I inject these patients with bacteria, they will get better. Mind you, these bacteria cause diseases, so they're toxic. So some of his early experiments weren't the most successful or the most ethical for that matter. But at that time, you could literally inject toxin into patients, and no one would say anything. I think regulatory bodies would have a lot to say about that nowadays. But yeah, he actually developed like a cocktail of bacteria, and he tried many, and he ended up with this cocktail of these two streptococci, two different types of bacteria, and then he mixed them, and it was called Colley's soup, or as we know it today, Colley's toxin. And he injected this Colley's toxin into patients that actually got decent success. He actually was able to, and I'm sorry this is hidden behind, he was actually able to successfully treat many sarcoma patients. He had about 60% cure rate at that point, which is pretty good for the time. However, a lot of people died of toxicity from the bacteria itself as well. And I think the biggest problem he had was that he could not explain why it worked. Like if you remember, back in the 1800s, nobody knew that there was an immune system. Like research was not what it is today, and it was just the beginning of that. It was on pure observation that he discovered that he could activate the body's immune system, which is what he was doing by injecting these bacteria, these toxins in, was basically activating our own immune system. And as a result of that, the immune system gets amped up and is also then able to recognize the cancer that it wasn't able to recognize before. And yeah, like his research was like, I think, the basis of all of the the immunotherapy that we have today, and really is considered the father of immunotherapy. But yeah, like nowadays you can't just go about injecting toxins into people. Having said that, we've learned a lot from what he's done, and they are treatments, and I will describe them later, using viruses that actually the principles are very similar to what Corley's doing. We just have a better understanding of the process and how to how to inactivate it so that the patient doesn't get sick. So today I'm going to talk about 4 new emerging immunotherapies, and many of you probably heard of some of these. A lot of them have been approved for the clinic. Checkpoint inhibitors were approved. There's a couple of checkpoint inhibitors. The first one was approved in 2011, the second one in 2016. Oncolytic viruses, and this is what I'm going to describe as being fairly similar to what Coley was trying to do at the time, was approved in 2015, and CAR T-cell therapy, the first CAR T-cell therapy, was approved in 2017. And there's active research going on in many areas. There's a lot of also immune molecules that have been approved for use in the clinic in patients. Those molecules I'm not going to describe today, but if you, we can talk a little bit about it at the end if anyone's interested. But those more molecules that you inject into the patients, and they go interact with the immune cells in the patient and help activate the immune system. A lot of the immunotherapies I'm describing today are cellular, so they're based on some sort of cell or the other. So the first one, and I think this is one that many of you might have heard of, is called checkpoint inhibitors. It was approved, as I said, in 2011, 2016. And the 2 key scientists who kind of led some of these key discoveries got the Nobel Prize in medicine for their discovery of these molecules in 2018. So normally, and we go back to that video where we were talking about the immune system functioning. Normally, these searcher immune cells, these phagocytes that we were talking about, that go search around for any pathogens, they go search around, and they discover a cancer cell, and then they're like, oh, you're off, something's off with you. In this case, the cancer cell is demonstrating some of the telltale signs of not being a normal cell. And the phagocyte, the searcher immune cell, it then goes and takes that signal, that information, and tells that to the killer immune cell, the lymphocytes that we were talking about earlier, that in particular the T cells, and it tells the T cells, okay, I found this cancer cell. Have a look, and these, these lines that I'm showing here are basically the signaling between the phagocytes and the T cells. So this phagocyte is telling the T cell, look at this, I found this molecule on this cell. So this cancer cell is showing this telltale sign. You need to go look for this telltale sign. So the telltale sign here is that green dot there. So showing it to the T cell, and the T cell is like, okay, I see it. I'm going to go find what's wrong and get rid of it. So then this T cell, the killer immune cell, goes around the body, looks for the cancer cell, finds it, because that's what T cells do. They're very good at doing that. Once they get activated, they'll make many of them, and they'll scour the whole body, and they'll be like, oh, there's that sign that the phagocyte was telling me about. And now it knows, it's like, okay, I'm going to have to kill this cell. But then the problem is the cancer cell is expressing that suppressive signal, which is one of the cancer's way of evading the immune system that I was talking about earlier. So it's this stop signal here. So it's this molecule, the cancer cell is showing the killer immune cell this molecule, and that is basically the cancer cell telling the immune cell, stop, you cannot kill me. And the killer immune cell is like, okay, it becomes inactive. It's like a negative signal to the killer immune cell, and it stops working. So what checkpoint inhibitors do is they are molecules that go in and they break this stop signal. They break that handshake that the cancer cell is doing to the killer immune cell that says, oh, you don't have to kill me, I'm fine. It breaks that handshake, and now the killer immune cell is like free to kill the cancer cells. So it's a very smart way of reactivating the T cell and bypassing that suppressive signal that the cancer cell is providing to stop the killer immune cell from functioning. So that one of the most I think famous, well-known cases of the use of checkpoint inhibitors is former U.S. President Jimmy Carter. He, in 2015, when he was 91, announced he had stage 4 melanoma, which had spread to his liver and his brain. And he was treated with radiation and the checkpoint inhibitor Keytruda. And he is now cancer-free and no longer needs treatment, and he continues to be monitored. Keytruda is basically one of the checkpoint inhibitors, basically one of the molecules that can break that signal. And scientists are continuing to identify more and more suppressive signals that the immune cell is getting from the cancer cell. So more and more suppressive signals that the cancer cell is showing the immune cell, and trying to find molecules like this to break it. So there's two checkpoint inhibitors that have been FDA approved and is in therapy at the moment, is being used in therapy at the moment. This one targets PD-1. And in this particular case for melanoma, the survival changes from months to at least 3 years. So it's actually decent, and people are trying to amplify the response by combining different types of immunotherapy with this. The second type of immunotherapy is called adoptive cell transfer. And this goes back to the T cell again, that lymphocyte that we were talking about earlier. The T cells, really the effectors of the immune system, right? They're the ones that are responsible for going and taking care of everything. So because in the immune environment, like in the tumor environment here, there's lots of immune cells, and these immune cells are all either being deprived of nutrients or being given suppressive signals by the cancer cells, so they don't work. So what scientists have done is they take some of these T cells from within. So they take a biopsy from within the cancer. Or when they resect the cancer, they take, they separate out the immune cells. And then what they do is in the lab, they actually activate these immune cells. So these immune cells have been suppressed. They take them out and they're like, okay, I'm going to boost you, give you a little activate boost so that you now can go over and get over that suppressive signal. And then they expand these cells. So basically here, they get the tumor, they dissociate it, break it up into little pieces so they get lots of different cells, take the right cell, the right immune cell, in this case the T cell, activate it, expand it, and put it back into the patient's body. So it's the patient's own cell, so the patient doesn't really reject it or anything, puts it back into the patient body, and now that these cells are amped up, they can go in and recognize and kill the cancer cells. And this adoptive cell transfer can be done with not just T cells, but any cells. Like people are exploring doing this with, the phagocytes as well, so that these phagocytes can be amped up, go in, recruit more T cells to the tumor environment. So there's a lot of research going on in this area. A very specific type of adoptive cell transfer is the CAR T cell. So this is our third immunotherapy. It is distinct because in the adoptive cell transfer, we only just boost it. We give like a chemical boost to these cells and expand them and put them in. In CAR T cell therapy, we're actually modifying your T cells genetically. In the backbone of CAR T cell therapy is again here T cells. Having said that, people are exploring using other immune cells for this purposes as well, but the approved ones at the moment are T cells. So in CAR T cell, what you do is you draw blood from the patient. Mostly this is for patients with different types of lymphomas, and you separate out the T cells. And then you use a virus, in this case, a retrovirus that's been inactivated, a disarmed virus, to genetically engineer these cells to express something on their surface. So these cells, they attach something genetically, they get these cells to produce something on its surface. That's called a CAR. Now, CAR is chimeric antigen receptor. I don't want you to stress about that name. What it does mean is normal T cells have natural versions of this. You're just giving your own body's T cells the ability to recognize the cancer cell because it doesn't have it, right? So you're basically attaching the recognition, like the recognition arm to the T cell, and then putting it back into the body. And now with this recognition arm, these cells can then go find the cancer cells, and the recognition arm will recognize one of the molecules on the surfaces of the cancer cells and then be like, oh, I've got to kill you. So in this case, we're actually genetically modifying the T cell so that it can specifically go and recognize something on the surface of the cancer cell. And it's very, very effective at killing them. So one of the cases I really like to bring up for CAR T cell therapy is actually probably one of the first few patients who got CAR T cell therapy. Her name is Emily Whitehead. She was a patient of Carl June, the professor, clinician who discovered CAR T cell therapy. She had acute lymphoblastic leukemia in 2012, and she got CAR T cell therapy. At that time, it was still, it was a clinical trial, and yeah, so he injected her, and because the immune system gets really, really activated, she got really, really high fever, and she got symptoms of what they call a cytokine storm, or cytokine release syndrome, which a lot of you might have heard in the context of COVID. A lot of patients, when they get COVID, their immune system reacts really, really strongly, and they get a cytokine storm, and that's what is actually causing a lot of hospitalizations of people. So when she got this CAR T cell therapy, her immune system got really, really amped up, and that caused her to get really sick. So of course she was really monitored. She was put on a ventilator for 2 weeks, and they kept a close watch on her, to try to make sure keep her safe. And she came through, and she's been cancer free for now, I think up to 8 years, which is amazing. And the really, really nice bit of this is her father actually was one of the people who, with her at his site, spoke up at the FDA approval hearing in the US for this therapy, and it was later approved in 2017. So that's a really remarkable story. And I think this is what drives a lot of scientists to continue to work in this area. And there's a lot of work in trying to make these therapies safer at the moment, and more effective, and to try to find different versions of this molecule that we can attach on to patients' T cells to help recognize the cancer. And I know I'm yabbing on, and I just want to make sure I leave some time for everyone to ask questions. But so this is my last immunotherapy that we're going to describe today, and these are called oncolytic viruses. Now, the word oncolytic, onco is the cancer, like oncology, and lytic means break. So literally, the name of this virus means cancer breaking, so cancer killing viruses. So these oncolytic viruses are able to, so this is kind of like what William Coley was trying to do. He was injecting bacteria into the body, and what that bacteria was doing was getting the immune response amped up against the bacteria, and then what happens is it then kills the cancer cells on the side because the immune response has become more acute. What these viruses do is they actually go into healthy cells, and they spare the healthy cells, because the healthy cell has antiviral mechanisms, so that the virus, it kills one cell, and then it doesn't translate to another cell, because there is, an actual process in the body that helps our cell warn other cells against viral infections. The oncolytic viruses, though, can affect cancer cells specifically, and because the cancer cell is it's changed some of its normal way of working, and it's trying to be very efficient in growing. It doesn't have these protective mechanisms. It doesn't deem it important because it doesn't think it's going to be infected by a virus. So basically, it's evolved to kind of, not have these viral protective mechanisms. So once these oncolytic viruses, and they're specific, not every virus will do this. There's a specific class of viruses, and there's a lot of research going on into making them more specific to cancer cells as well. Once these viruses, get into the tumor cell, they will replicate in the tumor cell, and then the tumor cells would die. This is what normal viruses do, when they infect the human body. But in a cancer cell, for this particular virus, in a healthy cell is fine, but a cancer cell is not. And then there is like a direct killing of cancer cells. And then what happens is, because this cell is dying, it's releasing signals into the immune system, and more viruses. So it causes inflammation, local inflammation, which basically then recruits more immune cells, and that will also help kill cancer. The viruses will then go infect more tumor cells, and it forms this nice positive feedback loop that causes the killing of the cancer. So the one oncolytic virus that is approved is called TVEC. And it's been approved in the US and in Europe, and it's been shown to be fairly effective alongside checkpoint inhibitors, especially in patients with melanoma. So in patients with melanoma, when you get both of these together, it's definitely shown to be more effective. So there's a lot of research going on into not just these kind of immunotherapies, other types, and into combining them, into trying to find gaps in the immune system where we're like, okay, if we put this together, it works well together and helps get rid of the cancer. So where one of them might not, two of them might work really, really well. So even though we've made great strides in immunotherapy, it's been absolutely like fascinating to watch the field just grow exponentially, and our understanding is so much more than what it was even like 10 years ago. There's still lots of limitations, and you heard me talk about cytokine storm with that patient, Emily Whitehead, and there are still lots of cases like that immunologists and scientists and oncologists are still trying to understand, like, why does this happen to some patients and not others. So our level of scientific understanding still does need to increase, and there's still some cancers, like solid tumors, in particular brain cancers, that are very, very hard to treat. Brain cancer, there's like a blood-brain barrier. It's very hard for immune cells to get across that. So that becomes like a big hurdle for treatment. And these immunotherapies are definitely not cheap. We are lucky to be in a publicly funded healthcare system, but even then, like, that means that Health Canada has to really deliberate, like, before bringing each therapy on, because of the cost side of things. So there's a lot of research into going into trying to make it more efficient and trying to make it cheaper as well. So yeah, there's still a lot to do, and then of course, I think this requires a lot of cooperation, public understanding of how important it is, and how important it is to support science, and clinicians and researchers working with the general public, not just to educate, but also to do further research to help find better treatments for all of these intractable cancers. With that, I'm pretty much at the end. I just wanted to show you, this is the map of all the current immunotherapies that are going on in the world as of like 2 days ago. And there's definitely a lot. There's like 2,256 immunotherapy clinical trials going on. So that's really encouraging. So to me, like the hope is some of those will lead to approved therapies that are really, really effective. So there's certainly a lot of interest and research going on in this area. And yeah, these are some of the resources. So if you're interested in some ongoing clinical trials, there's a few places you can go and look at them. You can also go and just Google clinicaltrials.gov. It's a very quick way of finding clinical trials. But yeah, any of these websites are great resources as well. And yeah, that's if you have any, please feel free to visit us or communicate with us on social media. We have our website, lots of blogs on there, as Kinjo said, and we also tweet and have a Facebook page, and we haven't put it here, but we have a new Instagram page. We've become very trendy all of a sudden, and and yeah, like please, feel free to reach out to us on these platforms as well. And yeah, we'll happily take any questions now.
Thank you so much, Patty and Kinjal. That was a very informative presentation. So I think I'm just going to jump straight into the Q&A. So our first question, in the case of blood cancer, is there immunotherapy possibly or being studied?
Yes, so the CAR T cell therapy that I described earlier is actually most effective in blood cancers. So that is one area of research, because a lot of blood cancer cells have, the CD19 CAR that I was talking about, they have the CD19 surface marker. So it's easier to go, more accessible as well for that T cell. Once you inject it back into the patient, it goes straight into the bloodstream, right? So it's easier and more accessible. Having said that, that particular class of CAR is not effective for every blood cancer. And there's a lot of research going on into CAR T cell therapies to try to find different molecules so that it can be effective for other blood cancers. Yeah, and there's also other immunotherapies that are being explored for blood cancers as well, but the CAR T cell is probably the most exciting one.
Mm-hmm. Thank you for that. Okay, so I think while I don't see any more questions, I'm not sure if people are typing them, but in case you wanted to just provide, I guess, just a quick summary of, if you had to maybe list like the top 3, I guess, current findings or current trends in research that are being studied, maybe that would give people some direction on what they can expect in this field.
Yes, so that's a very good question. There's a lot, and I have to pick one. So okay, so this is kind of biased of me because I work on this cell type, so I really like it. So forgive me if I'm being biased. But one of the classes of phagocytes that we were talking about, there's a class of phagocytes called macrophages. And at the moment, there's a lot of interest on macrophages because in solid tumors especially, there's a lot of macrophages in the tumor environment. And people are like, okay, these macrophages are supposed to go find the cancer cell and then tell T cells that these are cancer cells. And then why are these macrophages just sitting there, not doing anything? What I want to say about the macrophages is they also have the ability to directly eat a cancer cell if they deem that the cancer cell is infectious. And it's not doing that. It's sitting there in the solid tumor, not doing anything. And people have, over the last, decade of research found that these macrophages, they call them as tumor-associated macrophages, are actually have been suppressed. So they have been suppressed by the tumor, like a lot of the cells in the system, and they are kind of ineffective. So there's a lot of interest in activating them, and then they found a molecule on the cancer cell, that a different molecule that the cancer cell is using as a suppressive signal. So there's a couple of therapies in that area that are being researched really heavily. One is like an antibody against this signal that the cancer cell is providing to the macrophages, so it goes in much like a checkpoint inhibitor. So they haven't termed it a checkpoint inhibitor yet, but it functions in a very similar way. It goes in, sits and captures that signal that the cancer cell is giving and being like, okay, stop. And then the macrophages are free. But then even if the macrophages are free, these macrophages are still suppressed. So you still have to go and activate them. So people are researching new molecules. And there's a lot of like molecules that can activate macrophages, and people are researching molecules that can activate macrophages. So there's new therapies where you inject cells in that are expressing these molecules, so they go straight to the tumor. They've been designed that way. They go and they recognize the tumor cells. They go straight to the tumor environment, go in, release this, molecule load that it's like, this kind of chemical payload that you've coded it to express. So it really releases these chemical payloads and then converts these inactive macrophages into active macrophages. And that activates the whole cycle of immune system. So not only do the macrophages start killing cancer cells, they also go get the lymphocytes in, and then the cancer gets destroyed. So yeah, so that I think is probably like two therapies, the cellular side of it and the molecular side of it as well. Like, so with a suppressive signal, but then putting in chemicals to change the environment within the solid tumor as well, I think is very exciting.
Thank you so much, Patty. So our next question is immunotherapy done in conjunction with other treatment types, for example, chemotherapy and radiation?
Yes, I mean, this is like, it's definitely a case by case basis that, it really does depend on the oncologist and the type of disease. But yeah, very often there will be surgery, there will be chemotherapy and even radiation alongside alongside immunotherapy. Yeah, very often, a lot of those initial treatments are done. And those are like the classic three, right? Like surgery, radiation, chemotherapy are done to reduce the mass of the major mass of the cancer, and then you can use immunotherapy. But there's also a lot of cases in which, in certain patients who have, they sequence so they do a biopsy and they do a molecular analysis of, they analyze your tumor cells, and they say, oh, your tumor cells, your cancer cells are actually expressing that signal, the suppressive signal that the checkpoint inhibitor is good for. So those patients directly get, kind of the checkpoint inhibitor, because that is going to be most effective for them.
Mm-hmm. Thank you for that. And so our next question, is there immunotherapy available for ovarian cancer?
I think, if I'm not wrong, the checkpoint inhibitor can be used for ovarian cancer, but I wouldn't quote me on that. Like, I'd have to go look it up. And I think a lot of the immunotherapies are very, not the CAR T cell. The CAR T cell is very much at the moment geared towards blood cancers, but other immunotherapies are very general. Like checkpoint inhibitors, can work if your cancer is expressing that molecule. It can work across all cancer types. So I think the best way is to like talk to your oncologist and see if there is, an immunotherapy clinical trial or approved, immunotherapy drug, a molecule or cellular therapy that is approved for particular type of cancer.
Thank you. And so I think I'm just going to try to get in one last question, seeing that it is almost 6:30, I think. Is there a timeline for the possible use and affordability of immunotherapy?
Oh, that's a big one. Wow. I think, I'm in industry now, and I'm just starting to see this push towards trying to make things more efficient and cheaper. Having said that, I don't think anyone has a timeline. I think we're all like, the clinical trial is that bottleneck, right? Like, and a lot of the research that we do up front don't even make it to there, because it has to work in the dish, in the mouse, or in whatever animal model, and then in a clinical trial, in healthy patients, to make sure it's safe before actually being administered to cancer patients. So the process takes years and years, and there is a big push to speed it up. But regulatory bodies as well, rightly so, don't want to let in any therapy. It has to be safe. First and foremost, do no harm, right? So I think that's my quick way of saying I don't have a timeline. I don't have a timeline that I know of that the field has, but there is so much research going on in trying to make everything cheaper, faster, and we as a field are progressing like exponentially every day. So yeah, like, stay tuned. I think it's onwards and upwards from here. So yeah, fingers crossed is what I'm saying. We're trying.
Mm-hmm. Thank you so much for that. So I just wanted to, I think with that, we'll wrap up for today, but I just wanted to extend heartfelt thank you to both Patty, you and Kinjal for coming on today with us to present on this topic on behalf of Riot, and for everyone who was able to join us today. So thank you so much for addressing this content. And we do have a couple more Riot Be Well talks coming up soon. I think the next one should be on October 22nd. I don't think it is on our website, but would be will be up there soon. So you feel free to check that out as well and register for that if you are interested in hearing more from Riot. So I think with that, just a thank you again to everybody, and I wish everybody a good evening, and for everyone to take care during this time. Thank you so much.
Thanks, Renita, and thank you, everyone. Take care.
Thanks, everyone.